Okay, I'd like to welcome everybody to Pediatric Grand Rounds, and I'm going to turn this over to our Vice Chair for Education, Dr. Sandrine Van Schaik, to introduce our speaker for today.
Thank you, Dr. Hirsch. It's my great pleasure to welcome our speaker for today, Dr. Mercedes Paredes. I was just saying to Dr. Paredes, we've known each other forever, although I don't really remember how and when we first met. But she is a fantastic physician scientist and a leader in her field. She's an associate professor in the UCSF Department of Neurology and in the Neuroscience, Developmental and Stem Cell Biology and Biomedical Science graduate programs. She's a native of LA and received her undergrad degree in biochemical sciences from Harvard University and then joined the UCSF Medical Scientist Training Program. She completed neurology residency and postdoctoral training at UCSF, where she studied the development and survival of cortical inhibitory neurons. Her lab focuses on identifying features of neuronal progenitor proliferation and migration that are unique to the gyrated brain, such as in humans, with an emphasis on the perinatal period. She's a practicing neurologist who serves epilepsy patients with neurodevelopmental disorders and is passionate about mentoring underrepresented in medicine individuals in careers in medicine, STEM, and neurology. Diversity within the Department of Neurology is an important mission for her. As co-chair of the Diversity Committee for the department, she worked to promote awareness and education regarding diversity issues in neurology clinical care and diversity among the staff of the department. Her lab offers teaching sessions to local San Francisco Unified School District high schools to enhance neurology awareness and potentially recruit new diverse members for the future of neurology and neuroscience. With this expertise, she's working with the neuroscience program on longitudinal outreach efforts. She serves on the MSTP Council to offer mentoring and support and serves on speaking panels at the yearly Chicano Health Education Dia de los Muertos conference run by the Latino Medical Student Association. Lastly, she serves on admission committees for the neurology residency program and the MSTP Council, granting her the opportunity to meet future clinician scientists and promote applicants of diverse backgrounds. She is going to tell us much more about what her lab is focusing on and what research she's been engaged in. And so I am going to hand it over to her. She is going to talk to us about postnatal cortical development. I didn't even know that that was a thing. So welcome, Dr. Paredes.
Thank you so much, Dr. Van Schaik. It's an honor, and I appreciate your and the committee, Grand Round Committee's invitation to be here. And as an adult neurologist, I'm especially excited to speak with this community. And with respect to understanding neurodevelopmental disorders, I in the epilepsy world see the long-term consequences that you see from the beginning. And I think there's a lot of shared interests, shared areas of work. And I'd like to really focus on trying to link the emergence of these neurodevelopmental disorders to our understanding of the normal human brain. So I'm excited to share some of the work that my lab has done today. Can everyone see my slides okay? I want to make sure that that's working. Great. So I have no conflicts of interest, and the agenda for today will be a few items. First, I'll talk a little bit about the definition of neurodevelopmental disorders and recent changes that have come to light and some of the ideas behind that. I'll discuss the genetics and neuroscience, our understanding of what underlies neurodevelopmental disorders. And then finally, I'm going to talk about my own lab's work, focusing on a very unique period of brain development, what I consider the black box of human developmental neuroscience, the early postnatal period, and potentially how it might link to the emergence of some of the neurodevelopmental disorders that we see later on, maybe in childhood or when the phenotypes emerge later. So neurodevelopmental disorders, as the name describes, it's trying to group conditions that we believe have underpinnings in disrupting the normal process of forming the brain. The diagnostic criteria is quite broad, and recently the DSM-5 used to categorize some of these conditions changed within the past 10 years. If the attempts to try to group conditions not to emerge oftentimes early in infancy, but through childhood and even as late as adolescence, where the underpinnings really are for the abnormal formation of the circuit. And that leads to the phenotypes that we see, changes in cognition, learning, and behaviors. Now, there's a lot of discussion about how these groupings are, which we will discuss. but there are shared features that I think really have been consistent. One is the fact that these are multifactorial. Many conditions, many insults can lead to the variety of disorders that we will discuss. Across various studies, we see that there's an increasing rate of diagnosis, limited treatments, and the cost of care that makes this a really high public health concern. And through all this, another shared feature that really has been highlighted across many, many studies is the predominance of male this phenotypes in male patients, where men are most commonly affected. Some studies suggesting up to 2 to even 3 times as much. And then finally, again, as the adult neurologist, I really am seeing the chronic course of many of these conditions. There's increase in life expectancy among some of these conditions. Just overall, that really starts to highlight some of the comorbidities that start to emerge, the unique features and health concerns that these patients have. With this change in DMS-5, a new term came out trying to create an umbrella of disorders of intellectual disability, creating new categories such as communication disorders, and also now bringing into this umbrella the conditions grouped under autism spectrum disorder. And that in particular has been under very significant debate. How broad and how narrow should these categories be? There's no discussion that the prevalence of this is actually quite high of all these developmental conditions, higher than we would have imagined. Across any of these conditions, the prevalence remains about 14%, depending on the study. And the studies are also affected, and the disparities are seen with respect to access of care, demographics, and those who are able to get care, and again, the gender differences with the male predominance really standing across many of these conditions. Unfortunately, there's no clear data on some of these factors in adult patients, and that's an active process that's really needed to be interrogated. But what's clear, and I think this population, this audience knows quite well, is the impact not just on the patients, but also on family members who are oftentimes the primary caregivers and siblings. And all this ties to really trying to understand what are the medical needs and how can we best support these patients. Now, when defining neurodevelopmental disorders, the DSM-5 tried to create, again, this really broad categorization to bring in many conditions that are felt to have developmental underpinnings. That includes the new term of intellectual disorders, communication disorders, which include conditions such as stuttering. They moved attention deficit hyperactivity disorder within this category, and also specific learning disorders, including dyslexia, dyscalculia, and other language disorders. Motor disorders and autism spectrum disorders were also grouped into this category. And that's raised a lot of questions with respect to, is this category broad, and what are really shared features that can help us with the diagnosis? And what are aspects that are quite different across these conditions that we need to keep in mind, even in keep grouping them together and trying to create a bigger category for support and network of resources to offer. I think also underlying this shift was this idea that we need to separate and use less, have less reliance of definitions such as the IQ quotient to categorize patients. That was actually frequently used in prior DSM-5 versions. Those categories had very significant limitations and biases in their scoring. And now there's been a call for, across all these conditions, all these modalities, a need for really deep neuropsychological and neurocognitive testing and measures to be able to incorporate things like executive function into our diagnoses. And also, I think underlying this idea is that many of these conditions, because we now start to see them more and more increasingly in the adult clinical settings, we have to understand, are there special or unique comorbidities that can emerge with age? One such feature that's gotten interest within increased interest in the world of neurology has been the association of neurodevelopmental conditions and dementia. One Canadian study actually postulated that the risk would be quite high, even 6 times higher in general to develop dementia than in the general population. But it's definitely, even though the numbers are still under debate, there's definitely been increasing consensus that adults with intellectual disability of some form are at higher risk of developing dementia. But one classic example that has been really gained a lot of attention has been the connection between trisomy 21 that leads to Down syndrome and dementia. In patients with trisomy 21, there's been identification of about 4 to 5 times of overexpression of amyloid precursor protein and increased levels of amyloid beta. These are pathological canonical markers that have been identified and seen relevant in Alzheimer's disease and really have been shown to have increasing presence and accumulation in Down syndrome patients. What's also interesting, aside from making this connection, is the fact that some of these patients can present with their phenotype in different factors, and that can make diagnosing really complicated. For example, in trisomy 21, the initial changes that are often noted often relate more towards frontal lobe dysfunction, changes in behavior, personality, disruption, as opposed to the classic language and memory deficits that we see in Alzheimer's patients, or that was typically connected. And oftentimes, the caregivers are the first to come in and bring their patients for concerns for just their general malaise and fatigue and subtle behavior changes. And again, that really creates a link and a delay in diagnosing. And while we might not have the best treatment yet for these neurodegenerative conditions, I think an early indicator and an early identification for these conditions and this association is important, because not only it allows us to better understand this link and diagnose earlier, but also to connect these patients with further and better support that they might have if they had appropriate diagnosis. So now the world of neurology is trying to create more standardized and specific testing, such as the questionnaire for people with learning disability to identify dementia and neurocognitive changes earlier and more accurately. Another case of comorbidity with respect to neurodevelopmental disorders is that with epilepsy. And most of this data that creates a relationship between epilepsy and NDDs comes from the world of ASD, or autism spectrum disorder, where it's been noted that about 30% of ASD patients will have a seizure disorder. And 9% of the patient, and this can be different depending on the spectrum of where the patient is with respect to ASD. Patients with no intellectual disability are at less risk, and the prevalence of seizure disorders is less than those patients who have intellectual disability. Patients who have both ASD and intellectual disability have higher risk for cognitive deficits and are more likely to have frontal lobe involvement earlier than later. Now, there's a flip side also that patients with epilepsy are increased risk for developing certain neurodevelopmental disorders. For example, they have up to 22 times increased odds of developing ASD. Though it's unclear which came first, did the emergence of seizures disrupt the cognitive circuitry and increase the risk for developing ASD? And that's an active area of interest. A challenge in all these settings is being able to get some of the routine studies and interpreting them to accurately diagnose these comorbidities. For example, some of the extended EEG analysis to get the physiological data can be challenging in certain cases. And also, oftentimes, there are high frequency of abnormalities of unknown significance that can emerge within patients without any of these conditions, and that can make any findings on EEG challenging to identify at times. But overall, there's really an increased desire to want to oftentimes even repeat some of the workup for these patients to really see the prevalence of the comorbidities and to better manage them together, both the phenotypes and some of the behavioral phenotypes that we see in these patients who have these dual comorbidities. Now, I often see these patients as they transition from the wonderful pediatric care that they've been receiving. And that's raised some interesting challenges for the adult neurologist or adult care provider. In fact, it's been so important in the epilepsy world that there were certain task forces, such as the Ontario Epilepsy Implementation Task Force, that converged in these past several years to try to create better guidelines and protocols on how do we best make this transition to the adult clinical setting. And in one study that this task force ran, they saw that up to 2/3 of patients, young adults who actually had active epilepsy, did not have a formal transition and had to themselves re-identify and reconnect to adult, both just general care and neurological care. So that creates a big gap of knowledge in their clinical care and their clinical history and limiting their management. Now, in the adult side world, when we receive these patients, it generates a lot of questions that we have to create a better, I think, understanding for what is the natural history of this patient individual that we're receiving, and how can we really focus on some of these transition points that are so crucial. We often have to involve the caregiver and guardian, and I know I tap into my pediatric colleagues for advice on how to best do this. We have to think about, for many of these patients, they've often had their driving limited early on. They're getting, and those who are able to get their driving restrictions or driving privileges later in life. So how do we navigate that? There's also the impact of any of the cognitive deficits or behavioral changes that they have on their education, on their school or work environment. And then other topics such as sexual activity, use of birth control have really become an important feature as part of the introduction to the adult world for these patients. And lastly, as I touched on, is there a need sometimes to take some of these patients and repeat some of their workup? During this change in puberty, are there things on the EEG that might emerge that we need to be either regain knowledge or obtain a new baseline for these individuals? And lastly, we are receiving these patients during a very critical period in their adolescence, where we know that there's fortunately a high rate of medication adherence. And in the case of epilepsy, that can have really, really important ramifications. There's also high rates of depression, anxiety, and other mood disorder comorbidities that we need to be aware of and be able to work together with the individual and with their other providers to best manage. And then lastly, there's a lot of impact on their education. There's, for many of those who want to seek higher education, this is a critical period. And we need to figure out as a community how to better prepare institutions to support them, increase faculty awareness of the unique needs that these patients might have, and to be able to help those institutions better target their efforts for supporting them. All this together really shows that this lack of diagnostic criteria, the imprecise still nature of how do we define a neurodevelopmental disorder really has huge, huge consequences where many needs can go unmet. Especially as these patients are entering the adult world, a lot of the support that they receive and services changes or even lost. And how do we get them access to the adult version? There's also a lack of recognition. There's a lot of stigma even today regarding diagnosis of these conditions. And so being able to better define them and create awareness of them, I think is so important to be able to provide these patients appropriate support. And I think the past 2 years of pandemic has really shown us that these patients really uniquely lack the ability to access resources. They might not be able to cognitively access them easily, or their care providers might have limitations, whether it's geography. And how do we implement tools and technology such as telehealth to be able to access all the patients that need us? And I'll end on this idea that, well, it's a very broad category that has emerged from the DSM-5 definition of neurodevelopmental disorders and the idea of having neurodevelopmental disability. The highlight is that the goal, while staying broad, is to help establish diagnosis, be able to meet the needs and connect as many patients as possible to the resources that they can get to by having this diagnosis. The term disability, the organizers highlighted, was a way to show the impact that this condition has, not just on the patient, but on caregivers and providers. And the term neurodevelopmental was a way to emphasize the biological basis. So I think what really stands out is that we still have so many questions with regards to this. And while we have really the suggestions in the preliminary data to really highlight the developmental underpinnings, there's still many, many gaps in our knowledge. And we need to fill in these gaps to both not just develop better treatments, but to better define, categorize, and offer the support, the additional support that we can for these patients. And so to do that, I think we need to go back to the bench, go back to the basic science, and ask the fundamental questions of how these conditions emerge. And there's been a lot of interest. I think advances in genetic tools and sequencing technology in the past several years have really expanded our understanding behind the genetics and the potential mechanisms that underlie so many of the neurodevelopmental disorders that we've talked about. And recent high throughput sequencing has suggested many genes as potential targets, both as the culprit for these conditions and potentially as therapeutic targets. But it's been a challenge. While family studies have reported increasing rates of concordance for neurodevelopmental disorders in twins and in family members, thus supporting the high level of heritability, There's still a lot of heterogeneity, and we definitely know that there are also non-genetic influences that can lead to the emergence of these conditions. Part of the challenge of understanding the genetics behind these disorders is the variable expressivity and the differences in the penetrance. The unclear nature of the inheritance also is underlined by the fact that not only are there germline mutations, so very early mutations that lead to these conditions, but there can also be later somatic mutations that are present in specific organs, such as the nervous tissue, that can lead to these conditions in a very focal fashion. One prominent example is the presence of mTORopathies, where components of the mTOR pathway, a signaling pathway so fundamental for cell growth and cell development, mutations in that and a variety of those components can lead to different conditions, from TSC or tuberous sclerosis complex to malformations of cortical development, such as cortical dysplasia or hemimegancephaly. And then within all this, we also are having increased awareness that there's significant contribution of de novo or new mutations, as well as copy number variants or CNVs that can make up so many of the cases that we see, up to even 30% of autism spectrum disorder cases. So there's still this huge gap, I think, in understanding neurodevelopmental disorders, taking it from the gene influences and those environmental influences that we are aware of, to the phenotypes and the clinical behaviors that we see in our patients. But then the gap in the neurobiology, where in the development, where in all those developmental cell populations and mechanisms are we injuring that can lead to these conditions? And so I think to really dive into this question, we really need to understand, how does normal brain development, particularly how does normal human brain development occur? Now I'm going to step back and talk a little bit about cortical development, in particular brain development, focusing on the prenatal period. And why? Well, the prenatal period or the fetal period is really the time period that most of development of the cortex takes place. And those steps take place across three kind of classic ways. Early on, it's through proliferation, divisions of progenitor cells. And these progenitor cells reside in the ventricular walls of the developing embryo brain. In this area, often referred to as a ventricular subventricular zone, is where stem cells reside and then generate the daughter cells, the developing young neurons that then migrate away from this neurogenic niche in the prenatal brain. Excitatory neurons migrate fairly locally to enter the developing cortical plate that's going to become the cortical layers. And inhibitory neurons, they're generated further away and have to have a long tangential migration to reach there, to ultimately then incorporate with the excitatory neurons and then organize themselves and integrate to form the actual functional circuit. Now, this process is fairly conserved across all species. And again, most of it does take place in the prenatal period. However, in studying now the developing human brain directly, we started to realize that this process has really expanded and that there are distinctive features that have emerged. For example, in the human brain, there are an additional population of progenitor cells that I've highlighted called outer progenitor or outer radial glial cells that reside away from the ventricular wall. And it's thought that their presence is a mechanism for how the human cortex has been able to generate the additional neurons, maybe even more the diverse set of neurons necessary for the larger brain. In addition, we're now understanding that the human brain undergoes 40 gestational weeks of development compared to some of the shorter periods in the animal models used. And while we've gotten foundational information from these animal models, this extended developmental trajectory and timeline really has implications. And some of the features of development that we thought were restricted to the prenatal period have really now started, we realize, actually bleed into later times after birth. And less is known about these steps that take place in the postnatal period. When thinking about this postnatal period, especially with regards to the cortex, the focus has been on the morphological maturation and myelination that takes place. However, recent studies are really suggesting that there are ongoing changes to the neuronal composition, even for several weeks after. Here I show you data from the Moynier lab in Germany that used a mouse line, a reporter line, where they had green fluorescent protein, or GFP, that labeled the specific subset of inhibitory neurons. And they showed that even for about 2, maybe even 3 weeks after birth, there was still migration that was taking place in short little spurts going to the cortical regions. So these neurons continue to migrate, and even a small subset continue to divide and migrate and reach out to modify the cortical circuit. Now, in the infant brain, this ongoing migration of inhibitory neurons has also been identified. Initially, this was work done by Sinai et al. From the Alvarez-Buya lab. They were looking at populations of inhibitory neurons that were going from the ventricular wall all the way to the olfactory bulb. Now, this population, stemming from the ventricular-subventricular zone, is known to contribute across other species to the ongoing generation of new neurons in the olfactory bulb throughout life. And this rostral migratory stream was indeed identified and found in the infant brain, here shown at 5 months. And this stream had many features that was identified in other models, including the presence of double cortin, which is a microtubule associated protein that is present in the cytoskeletal of migratory young neurons. So it's often used as a marker to identify and find these young neurons. And so the stream is filled with double cortin positive cells and ensheathed by GFAP positive astrocytic fibers. Now, interestingly, while this was present in the infant brain, this RMS stemming from the ventricular-subventricular zone in the human was transient and disappeared by 1 year of life. This also correlated with a decrease in stem cells in the subventricular zone of the ventricular wall. But interestingly, in looking for this RMS, Sinai et al. Identified that there was a branch, here identified by double cortin expression in green, that emanated from the RMS and targeted a very focal region in the prefrontal cortex. The stream of neurons was called the medial migratory stream, or MMS, and was present also transiently, just for within the first year of life. And interestingly, when they looked at other species, such as the mouse, an MMS was not identified, suggesting that it was really something unique to this larger brain, such as a human. I will say, I'll give a little bit of a teaser that the MMS might be present in other higher, quote, higher level species. Now, what about other areas that could be receiving interneurons from this subventricular zone in the infant brain? So in work that I and colleagues did, especially spearheaded by David James, a talented medical student, now a psychiatry resident at Stanford, we started to look at sections of the newborn brain from postmortem autopsy cases. Here I show you a sagittal section taken at 39 gestational weeks, or term. And in this section, we could see cell dense collections around the anterior body of the ventricle, the cavity here. And this is shown by Nissl staining. Now, these cells that surrounded that part of the ventricle were double cortin positive, here shown in green now. And these cells within these densities were very uniquely morphologically looked migratory to us. And also, when you look closely at this, were organized in a very complex fashion, where young neurons were either longitudinally at the ventricular wall, clustered around blood vessels, adjacent blood vessels, or organized into clusters of varying sizes entering the developing white matter. You can see that better here, where you can see some of those cells with very migratory features of a long leading process and small nucleus. Now, we wanted to get, those were coronal sections. We wanted to get a more global perspective of these cells. And to do that, we teamed up with Hosam Kim and Duong Su here from our pediatric neuroradiology department, colleagues there, who said, well, hey, let's try looking at some of your postmortem cases and do intact MRI or in vitro MRI imaging. And in doing that, we really were lucky that we were able to identify a T2 MRI sequence that matched the signal that we were seeing with our Nissl cell stains. And that matched with those double cortin young migratory neurons that we were seeing in our histological analysis. So this would allow us to visualize these migratory streams in the intact hemisphere. Now, having this approach, we were able to generate a 3D reconstruction of this collection, here highlighted in green, and showed that the structure formed a cap around the anterior head of the ventricle. The ventricle is shown here in light gray. And it really highlighted two things for us. One was the far-reaching nature of this cap, going from the most anterior tip of the lateral ventricle all the way as far back as the thalamus. It also showed us the anterior displacement of this population between ages of 34 gestational week and 2 months, suggesting that this population from the anterior body seemed to be entering or moving into anterior regions of the cortex, such as the prefrontal cortex. Given the shape and organization of these cell populations, we dubbed this whole stream the arc. Now, in data I won't show you, we were able to take some postmortem cases, generate live imaging data where we cultured these cells, labeled the cells, and watched these cells actually migrating in the postnatal environment to really show that these static images of these young migratory neurons and the way that we were analyzing them actually related to cells actively moving in the postnatal environment. Now, David next subsequently mapped the position of these double Cortin positive young migratory neurons, each thought representing a single migrating neuron, in coronal sections of brains taken from birth to 7 months of age. Now, in doing that, he confirmed that each cell had migratory morphology, and he showed that these cells disappeared within the 5 to 7 month period. It was, really for several months only after birth. But also, his mapping showed the transition of these cells from the walls of the ventricle and the developing white matter at birth into the overlying cortex at about 1.5 months, and then the emergence of these cells looking more mature at 3 months. Now, he mapped those cells and showed that the peak of these waves, at least entering these anterior cortical targets, the cingulate and the frontal lobe, was at around 1.5 months. When he quantified the total neuronal population, he had taken in UN, he showed that this influx of double Cortin positive cells that peaked at 1.5 months was followed by a peak of the neuronal population here at 3 months. So showing that relationship of this entry of these cells to changes in the composition of the neuronal population, at least in this target region of the cingulate. Lastly, we showed that these cells were inhibitory in nature, given their expression of inhibitory protein markers, including GAD67, which is glutamate decarboxylase, which makes GABA, and the expression of markers associated with different interneuron subtypes, including calbindin, somatostatin, and neuropeptide Y. Now, with this really wanting to focus and better understand the composition and nature of these cells, we understood that we had to find an additional system to better interrogate these populations, given the rarity and preciousness of these term and neonatal samples. So my team turned to complement these human studies with a gyrencephalic model, the piglet. Now, the piglet brain we chose not only because it's also gyrated, but there are other features that are shared with the human, including the fact that both humans and pigs have an increase in brain size that begins in the 3rd trimester. And MRI studies on the domestic pig have shown that there is an increase in brain volume during the postnatal period that closely resembles that seen in humans. So Jae-Yoon Kim, a talented postdoc, and Kaylin Sandoval, a wonderful student in the lab, took on the challenge to ask, is the arc structure conserved in the piglet brain? And how similar is it to the human? And using the piglet brain, can we observe additional migratory streams in the postnatal cortex? And what are the molecular and functional differences between these streams? Now, cutting to the chase, I will say that indeed, the piglet brain has an arc structure. And we clearly saw, even initially, using missile stains to identify cells. And we could see these cell densities around the edge of the lateral ventricle, the cavity here, that was reminiscent of what we observed in the human. And these cells were double Cortin positive. You compare the human to the postnatal day pig here at postnatal day 0. And these cells had the organized tiered structure where they were clustered around the ventricular wall, dispersed around blood vessels, and then clustered as they entered the developing cortex. Now, we were really fascinated that the piglet was such a faithful model for this, and we were curious and actually expected that the non-human primate would also have an ARC equivalent. Now, for this work, we had teamed up with Angela Robert from the University of Cambridge. We had started to work with her to look at late migration of interneurons in the marmoset brain. She and her team used a marmoset as a model for mood disorders, particularly focusing on area 25 in the marmoset, which is equivalent to the cingulate target, the cingulate gyrus, which, if you recall, is a major target for ARC population. But interestingly, the marmoset brain, here I show you an example of one at postnatal day 1, lacked double Cortin positive cells in those cell clusters at the edge of the ventricular wall, with most of the double Cortin positive cells really tightly lined the wall. But we didn't see any dispersed cells entering the cingulate gyrus, like as in the ARC. We didn't identify a marmoset MMS, but only could find the RMS, which we could see here, which we had seen across in the human and other species as well. Now, stepping back and looking back at these gross anatomical images, we were reminded that the marmoset is actually a lissencephalic, more specifically, a secondarily lissencephalic model, where the only cell size is that present in separating the temporal lobe, a small temporal lobe, to the rest of the cortex. So this was fascinating, and Jae-young really wanted to understand and quantify this relationship between the ARC and gyrification. So to do that, she first began by creating an ARC ratio. When she did, she took coronal sections and measured the area, here shown in green, that was filled with those double Cortin positive young migratory neurons across the wall there. And she divided that area across the total area of that cortical section. And she generated an ARC area ratio for the human, piglet, and marmoset brains at equivalent perinatal ages. And what she saw was that the ARC ratio was highest in the human and nearly zero, very small, in the marmoset. Next, she generated a gyrification index, where she measured the inner length, here shown in red, which is the total length perimeter of the cortex, cortical surface, including the infoldings, and divided that by the outer length, here in cyan, which is the perimeter of that cortical surface. This is similar to analyses done in MRI imaging. And what she could see was that when she mapped and then quantified the relationship of the gyrification index to the ARC ratio across human, piglet, and marmoset, and she was also able to identify a data set to do these measurements from the macaque brain. She saw that the human, piglet, and macaque, all gyrencephalic brains, it had a very tight relationship between the gyrencephalic index and ARC ratio. This was missing in the marmoset. So really showing this tight relationship between those two features, the presence of the ARC and gyrification. I'll just say that Jae-young then went on to measure the presence of this ARC, and she did that between embryonic day 89 to postnatal day 28, and showed that even during this time when the brain is still increasing in total size, the ARC volume was actually fairly constant and didn't decline until postnatal day 28. So this matches up to the decline that we saw between the 3 to 6 month period in the human brain. Jae-young then showed that these cells were, as in the human, inhibitory in nature, given their expression of GABA and expression of VLX2, which is an interneuron progenitor cell transcription factor. Now, Jae-young and Caitlin wanted to understand what were the origins of these inhibitory neurons that they were seeing. Now, mouse models have shown us that inhibitory neurons come from the ganglionic eminence. These are ventral regions in the embryonic brain. Here I show you coronal sections of a mid-gestation mouse brain. And you can see the anatomic landmarks that separate the subregions of the ganglionic eminence, the medial from the lateral ganglionic eminence, and then as you move posteriorly, the caudal ganglionic eminence. These regions also have unique expression patterns for transcriptional factors that are used to identify these regions and are important for the differentiation and maturation of cells that originate from here. Factors such as NKX2.1 for MGE and CUPTIA2 and SP8 for the CG. This distinction of the subganglionic eminence is important because each of these subregions generates different kinds of interneurons. The LGE, or lateral ganglionic eminence, makes olfactory bulb neurons, And the medial and caudal ganglionic eminences make up most of the cortical inhibitory neurons in the brain. Now, in the mouse brain, it's been shown that the MGE makes up about 2/3 of the total inhibitory neuron population. And this specifically makes up unique subtypes of inhibitory neurons, the parvalbumin and somatostatin subtypes. And these have been implicated in many neurological conditions, including epilepsy. Now, the CGE in the mouse makes up a smaller proportion, and they make specifically the VIP and reelin inhibitory subtypes, and less is known about this subpopulation. But interestingly, in the ARC, when Jae-Young and Caitlin looked for the expression of transcription factors from the MGE, the CGE, or SP8, which is a transcription factor for the CG-LGE, they saw that the MGE actually is a small proportion of the double Cortin positive cells present in both the ARC and in the streams migrating away, and that the CGE and LGE, as identified by COOP-TF2 and SP8, were the most predominant population seen. Quantification of these populations across the different tiers or layers of the ARC really showed that this was consistent throughout all the areas that were populated by these young migratory neurons. Now, they compared the postnatal day piglet to the term human in equivalent areas of the ARC in each of these two species. And they showed that this predominance of the CG-LGA populations was present in both. But there was a unique difference where the piglet had more representation of the SP8 positive population, as opposed to the human, where they had both a combination of SP8 and COOP-TF2 positive interneuron subtypes, suggesting, while there were shared features, some species differences that we need to keep in mind. Caitlin, I wanted to really take advantage and leverage the piglet brain to map all the postnatally migrating neurons that were present. And she did that in postnatal day 0 piglet. And here I show you coronal sections going from the anterior to most posterior level of the ventricle. And each dot represents a double Cortin young migratory cell. And Caitlin was able to identify the rostral migratory stream and the medial migratory stream, in addition to the ARC, with cells going to the cingulate and overlying frontal lobe regions. But interestingly, she also was able to identify cells that were targeting and migrating towards more ventral and lateral regions, so areas such as the piriform cortex and the temporal cortex. So this broad distribution was something that Jae-Young wanted to confirm and also actually see cells migrating in the postnatal environment. So what Jae-Young did was take postnatal day 0 piglet brains and generate organotypic slice cultures. Now, she took these sections of tissue, infected them with either a CMV-GFP adenovirus or a CAG-GFP AAV2 virus to visualize cells in real time. Here I show you an example, day 1 in culture of a section that was infected with the CMV-GFP adenovirus. Now, most of these cells at day 1 were at the ventricular wall within the ARC region. But by day 5, these cells started to disperse and go out into the neighboring areas away from the ARC, and these cells were double Cortin positive. Now at day 10, not only could she see, as you can see in area 2, more cells dispersing away from the ARC, but now she could start to visualize those streams entering area 1, which is the cingulate cortex, area 3, areas of the frontal lobe, similar to what we had seen before in our coronal sections, in our histological analysis. And in more posterior levels of analyses, she was able to see cells migrating in that temporal lobe stream. So all this confirming that not only can we now visualize these migration and their patterns, but also see the extensive nature and broad nature of these cells. To get a more granular understanding of what the composition of each of these populations was, Jae-Young implemented spatial transcriptomics, the ability to look at gene expression at a single cell level and retain anatomical information. She used RNA-scope, which is single molecule fluorescent in situ hybridization approach, to identify gene expression patterns here, the genes that she showed in different colors. And these were genes that she wanted to identify and correlate the double Cortin positive cells that she was seeing with the different transcription factors that would associate them with specific ganglionic eminence regions of interest. And when she did that, she compared the populations entering the cingulate cortex to the temporal cortex. And what she noted was, just even qualitatively, that there was much more heterogeneity between the populations entering the cingulate to those entering the temporal lobe. And that there was a much more predominance of the SP8 population, here shown in, its gene shown in cyan, with respect to that in the cingulate. These differences in those two populations are best visualized in the quantification in the heat maps here, where she compared double Cort and positive cell expression of these genes in the cingulate to the temporal cortex. And what I think jumps out, one, again, that heterogeneity, that cells, double Cort and positive cells in the cingulate were both MGE derived, as you could tell by math expression, but also a variety of markers that related it to the CGE, including PROX1, secretagogue, SP8 again, and COOP-TF2. Now, when you look at the temporal lobe, again, you could see that predominance of the SP8 positive population. And interestingly, we saw the expression of DLX2. Now recall that that is transcription factor seen in interneuron precursor or progenitor cells. And this expression here in the temporal population, as opposed to that not as highly expressed in the cingulate, suggested that maybe this temporal population was less mature than those populations entering the cingulate cortex. So Jae-Young then went back to the coronal sections of the piglet brain from postnatal day 2 and postnatal day 28. And she looked at the migratory cells, here again, double Corton in green, and saw that the double Corton, first that the DCX really again stayed close to the ventricular wall, which would be in this area. And that streams peaked their migration out from this arc area towards the cingulate at around postnatal day 16, but then were fairly depleted by postnatal day 28. This is in comparison to the temporal lobe, where Jae-Young saw the streams heading towards the temporal lobe at postnatal day 2, but even as late as postnatal day 28. So suggesting that migration into the temporal cortical regions outlasts other cortical targets. Now, Jae-Young is working with other lab members to return to the human brain. And indeed, now she's been able to identify that these novel or previously unidentified migratory streams entering the infant temporal cortex are truly present. Here showing a more posterior level of the ARC population and those migratory streams emanating out, and then streams entering the temporal lobe. And she's really trying to focus now on particular specific targets going to different parts of the temporal lobe. So the piglet brain has similar ARC structure to that was identified in the human brain. And comparative studies, including non-human primates, shows that there's a really deep connection between the presence of the ARC and gyrification. Now, what is identity of these migratory neurons in the ARC? As in the human, they seem to be inhibitory in nature. And the piglet brain really allowed us to see that these postnatal waves of migration are predominantly CGE-derived, LGE-derived. So unique and really less understood interneuron population that really is now, I think, gaining attention. And that while there's a similarity of the CGE origins, there are subtle species differences that we should be aware of. Lastly, use of the piglet really helped us not only to identify the populations now entering that we had not known before, entering the temporal lobe, but also they have been able to show us that there is this diversity, that the populations entering different cortical targets aren't necessarily similar, and that the developmental timelines for these also might not be shared or might be staggered. And I'd like to end this part by saying that this idea of late incorporation of inhibitory neurons into the cortex is really intimately connected to the neuroanatomy, the presence of this ARC structure that is amplified in gyrencephalic brains. And that this leads to the emergence of a diverse set of what I call late migration into different cortical regions. And in particular, areas that have higher cognitive functions, areas of the temporal cortex and prefrontal lobe. And that these two areas, while they might share certain features, such as the predominance of the CGE molecular phenotype, but there are distinct patterns and distinct temporal patterns. And that this might offer a cellular mechanism to create regional specificity to plasticity and functional acquisition in these gyrencephalic brains. And I think that needs to be further interrogated. Now, the identification of those postnatal cortical migratory pathways began with really the idea of wanting to interrogate neurogenetic niche in the adult brain. And we saw how the walls of the ventricle that contain the ventricular subventricular zone, or V-SVZ, contributes to these late waves of migration. But that then in the human, they disappear during early infancy. Now, there is a second niche, the subgranular zone, that resides in the hippocampus, a brain region fundamental for learning and memory. So in the last few minutes that I have, I want to share some preliminary data of our work, trying to understand the emergence of this niche in the human brain. Now, quickly, imagine there's so many implications for the idea of having neurogenesis in the hippocampus. Factors that affect neurogenesis in this region include very clinically relevant features such as seizure disorder, exercise activity level, exposure to antidepressants, and altering neurogenesis in this region. And these animal models have shown changes in mood, regulation, and cognitive function. And obviously, the therapeutic implications of having newborn cells throughout life are really exciting and fundamental. But how does this translate to what's happening in the human? And I'll say that while in the mammalian hippocampus, and particularly that dentate gyrus, does harbor a stem cell population throughout life, increasingly data, including those from my own group and collaborators, are suggesting that young neurons and neural stem cells, neurogenic populations, are being depleted from this region during childhood. Here I show you a double Cortin positive cell that classically should look very migratory, actually looks more mature, like in the adolescent hippocampus dentate gyrus. And when we map to neurogenic populations, here I show you our mapping of double Cortin positive cells in yellow, we see that this population disappears by childhood and is gone after adolescence. And so Oyer Pastor and Hannah Lambing in the lab have really wanted to tackle the idea of, is there neurogenesis late in the human hippocampus from a developmental perspective by asking, how is this niche first established in development? To do that, Oyer and his team went back to analyze prenatal hippocampal samples. And here he shows you samples from 14 to 22 gestational weeks. And using Nestin in green to identify progenitor cells, we can see the connection between the ventricular wall, here identified and termed as the dentate neuroepithelium, or DNE, and that connecting to the developing dentate gyrus and developing granule cell layer. Now, this is present even at 18 gestational weeks, but you can see a breakdown of this connection between the ventricular wall and the developing dentate gyrus by 22 gestational weeks. Now, when you go back and focus in on the ventricular wall, you see that this area is filled with Nestin positive cells and TBR2 positive cells. This is still the case at 18 gestational weeks. But again, by 22 gestational weeks, this area really seems to be depleted by Nestin positive cells and those TBR2 positive intermediate progenitors. Now, when you focus in on the developing dentate gyrus and that granule cell layer in this region, we see that between 14 and 18 gestational weeks, the secondary neurogenic niche is emerging with better organization of fibers, Nestin positive fibers coming from the progenitor cells. And that by 22 gestational weeks, you see a beautiful organized fiber connecting the hilus that is below in the GCL, where the intermediate progenitor cells reside, in the outer part of the dentate gyrus. Now, fast forward to 30 gestational weeks, we see that progenitor cells have organized and formed a scaffold between the hilus and the granule cell layer, and TBR2 positive intermediate progenitor cells are really intimately related here to this scaffolding. But overall, the matrices in the other areas have really started to decline. And this whole organization and cytoarchitecture is more reminiscent of a juvenile rodent hippocampus and dentate gyrus. Now, 3 weeks later, we can see that the scaffold now has vastly diminished, as you can see here by the loss of Nestin positive processes and the decline in TBR2 positive progenitor populations. Finally, to try to connect all those changes that we're seeing with those neurogenic populations, Oyer has taken to use single nuclei sequencing, where he has taken samples from gestational week 9 to 38 years of age. isolated nuclei, run them through RNA sequencing, and then use bioinformatics to compare gene expression patterns at the single cell level and group these cells according to shared gene expression patterns. And I'll just end on the teaser that when he's done that, and this is done in collaboration with Li Wang in the Kriegstein lab, he's been able to identify all the subpopulations of interest, but in particular, a population that I highlight here of progenitor cells with astrocytes. And so he really wanted to interrogate the nature of those progenitor populations. So he pulled those cells out and reanalyzed them. And was able to identify the diversity in the subgroups of these progenitor populations more clearly and identify that there is a specific early progenitor population present only at 9 and 10 gestational weeks, and that the subsequent progenitor cells really separated into two progenitor subgroups that were only really present after gestational week 15 and then disappeared by 38 weeks of age. That these progenitor populations appear to have a distinct age-dependent trajectory that we're now digging into. Now that we have these subgroups, Warrior is going to try to identify what are the genes that differentiate these early and late progenitors, and what are potential cell molecular pathways that regulate the emergence and then disappearance of these progenitor populations. So I want to end this part by just highlighting that, again, just as we saw in the ventricular wall, young neurons and neural stem cells are depleted during childhood. But actually in the human hippocampus, progenitor populations are already organized and were quite mature quite early, by 3rd trimester. And that this area contains a diverse population of progenitor cells that have temporal specificity. We hope that we can, through these studies, highlight, are there dormant or quiescent stem cells that are present in the infant or childhood period, ones that could be reactivated? And how does the change of these progenitor populations relate to plasticity? And I'll just end on, I think overall, these studies on the postnatal cortex highlight the need to better understand and better find ways to study the human brain, to identify how our stem cells and how our developmental processes maintained in the infant brain, and how does disrupting these normal processes in populations lead to the emergence of neurological and psychiatric conditions. And I hope that the piglet brain not only offers us another model to interrogate the molecular composition, but also a way to look at the functional outcomes of disrupting these processes. And this would allow us now to connect genes and environment to phenotypes. And not only understand the fetal and perinatal windows of vulnerability, but potentially even identify new windows for therapeutic information, even after birth. And I hope this gives you 1 or 2 more minutes for questions. And I want to thank you for your attention. And I want to thank all the collaborators for this work.
Thank you so much, Dr. Paredes. I just learned so much, really very interesting talk. We have exactly 1 minute for questions, and there's one question from Dr. Lustig that says, can you talk about the differences in neuropathology that occur with hypoxia versus hypoglycemia, and whether the difference can explain the role of mitochondria in normal neuronal development vis-a-vis PI3K, AMPK, and mTOR?
Thank you for that question. We haven't looked in hypoglycemia, and we'll say that there's been collaborators who started to look at hypoxia, and that hypoxia does seem to decrease the number of those young migratory neurons present in areas that we think are related to the ARC. So I do think that there's a connection there. And yes, I think it'd be fascinating to think about what are the metabolic changes that are brought on, the requirements just to maintain this late wave of migration, and how conditions like hypoxia might push and create vulnerabilities in that. But I think those are active areas of interest.
And I want to be cognizant of the time. It's 1 o'clock. If you have another minute, then Dr. Rubinus is asking, she's saying, fascinating presentation. As a neonatologist, I was wondering if you could comment on any work done to elucidate how the ARC and postnatal neuronal migration patterns differ at different stages of premature birth, and how this influences the increased rates of ASD and other developmental disorders we see in children who were born preterm.
That's a great question. One aspect is we are careful in our samples to try to not use premature cases of prematurity, because we are concerned, MRI data has suggested that there are definitely global volumes changes that emerge from that. So we want to be careful of that. The flip side is we hope that in the future, we can access some of those cases that we obtained that come from premature patient populations to look at this. And even now that we can potentially identify some of these populations, at least for a short window, using MRI, I think that'd be a fascinating question, because we don't know. We just haven't looked at that, but I think that would be a very beautiful, specific question to ask. If anyone has questions, I'm happy to also answer them by email. Thank you for, I know it went a little bit over time over the question period.
No worries whatsoever. Thanks again for a fantastic presentation, and we see everybody next week on Grand Rounds. Have a good afternoon.
Thank you, everyone.